and entry into the cell. So before starting, I want to acknowledge one thing that we already studied. There's some glycoproteins on viral cell surface and also on the host cell surface. Those glycoproteins are receptor. They interact to each other, and their interactions help the viral attachment of the host cell, and then the virus can easily enter into the host cell. And the purpose of virus is to induce its genome into the host cell and replicate it. Okay, so uh, the aim of the virus is to replicate itself, and to achieve this aim it needs to enter into the host cell. In general, the process of virus replication can be broken down into 7 general steps. One is attachment to the virion cell, attachment of a virion to a cell, entry into the host cell, transcription of virus gene into mRNAs, translation of virus mRNAs into viral proteins, and genome replication, and assembly of proteins and genomes into virions, and exit of the virions from the cell. Those are 7 general processes of viral replications. Uh, but all of the 7 steps are not relevant to all the viruses. The steps do not always occur in the same order, or there might be additional steps also. So in later stages of replications, several steps occur concurrently. That means transcription, translation, replications, virion assembly, and exit can all be in progress at the same time. And uh, and to infect animal and bacterial host, viruses need to cross the bounding membrane or wall of the host cell. Most plant viruses are delivered directly into the cell by a vector. So first thing is need to cross the bounding membrane or wall. So bounding membrane is cell membrane for the animal cells and cell wall for the host cell. So here cell receptors and co-receptors plays very important role. A virion uses one or more of its surface protein, mostly called glycoprotein, to attach to a specific molecules on the surface of the host. And these cellular molecules on the surface of a host cell are known receptors, usually glycoproteins, with a wide range of functions that include acting as receptors for chemokines and growth factors. That means those glycoprotein receptors is not just for viral attachment. Basically, those glycoproteins expressed on the host cell for host cell purpose, to aid with chemokine actions, growth factors actions. So basically, the virus attack the receptor. Those are chemokine receptor or growth factor receptor of host cell, and mediating a cell to cell contact and addition. Some viruses need to bind to a second type of cell surface molecule on the host known as co-receptor. And and binding to receptor causes a conformational change in the viral protein that enables it to bind with the co-receptor. So do you know what are the changes, conformational changes of a protein that usually we know when a substrate binds with the enzymes, there should have some conformational change like that. So when a substrate or when a ligand binds with the receptor, that induce a conformational change, and that helps the further binding with the co-receptor, and that helps finally to attach uh, the viral particle with the host cell surface. Okay, so here some cell receptor and co-receptors for this is non-envelope, non-envelope, envelope, 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 and envelope. Those are the specific viruses. So for non-envelope rhinoviruses, cell surface receptors are intracellular adhesion molecule LDL receptor. For poliovirus receptor CD155, human immunodeficiency virus. These are envelope virus where you can find the CD4, CCR5, CXCR5 co-receptor as a co-receptor, and for influenza you will get sialic acid. For other SARS-CoV, you can see angiotensin converting enzyme 2, and for mumps virus you can see trisaccharide containing sialic acid, and measles you can see CD4. So most of the cases you can see the receptors are the receptors are immune system receptor. That means when our that means that the different factor that bind with the receptor in our body that trigger the immune system of our body, those receptors basically interact with the virus. So you can see here CD, angiotensin, sialic acid. Okay, and the remaining part you can see also here all the receptors here belongs to, not all, most of the receptors belongs to immune system. So mumps virus trisaccharide containing sialic acid, CD14 measles mumps, and rubella virus MOG. So there are different types of receptors. So what we learned, that means before entering the viral attachment is very important, and where receptors and co-receptors plays very important role for viral attachment. So each virion has a multiple site that can bind to receptor. That means you can see antibody antigen epitope. So like epitope, viral virions has multiple site on its surface that can react with the receptors, and virus attachment site on non-enveloped virus are on the capsid surface or on a specialized structure that we already studied, fibers, knobs, therefore spikes. Okay, virus attachment site of enveloped viruses are on the surface of glycoproteins. That means the receptors are glycoproteins. Some virions surface proteins of attachment site also able to bind RBC and cause clamp hemagglutination. So we studied in the last previous classes, that is the influenza viruses has hemagglutinin that can interact with glycoprotein expressed on the RBC and cause the agglutination of the RBC. Okay, so this is called hemagglutination. And the tropism, that means the particular direction in response to extracellular stimuli of the virus, is determined by which cell on the body express the cell surface receptor. The two important things. While viruses are tissue specific, there are two reasons. One, viruses always are looking for their favorable environment. So when they found the favorable environment, they will bind there. So each tissue provides a favorable environment for each types of virus. Favorable environment means expressing receptor so that this receptor can be recognized by the virus. Okay, and also some cells express some chemicals, release some chemicals, and virus attract toward those chemicals, and this is called tropism. Because of those, the viruses are tissue specific, and virus can bind or attach in particular tissues in our body. So these are structure of different receptors and co-receptors, and most of them you can see here is a IZ-like domain, 1, 2, 3, 4, 1, 2. This is IZ-like domain. Immunoglobulin-like domain, 1, 2, 3, and those are disulfide bond. So you know the cysteine is a amino acid that has a sulfhydryl group SH that can bind with another cysteine molecules to produce this disulfide bond. And immunoglobulin-like domains are linked together by this immunoglobulin domains are created for this disulfide bond. Okay, so most domains are stabilized by disulfide bond, and virus binding site is located in the outermost domain. So those are cell surface receptor where virus can interact. This is CD4 receptor, and this also work for HIV virus. So there are three domains. One is extracellular, another is intracellular, another is transmembrane. So many receptors in the plasma membranes are coated with inner surface with or one of the proteins of clathrin and clavulin. তাহলে কি virus binding sites is located in the outermost domain here, and those receptors in the plasma membrane are coated with inner surface with one of the proteins called clathrin or clavulin. Look at here. And these receptors had intercellular part. Those intercellular part is coated with some specific types of protein. Those proteins called clathrin and clavulin. So those clathrin and clavulins are responsible for production of clathrin coated peat, where the cell can trap the virus and can enter into the cell. And in case of clavulin protein, they can produce a cavule like peat, that means a cave, so that the virus can engulf in the host cell. Those are two important parts. So we will study details in the next part. So those are the co-receptors or receptors expressed by the host cell to interact with different purpose, but viruses uses those receptors to enter into the host cell. Okay, so here you can see this is CXCR receptor, CC5 receptor, and CD4 receptor where HIV can interfere. Cells are not expressing this receptor for HIV or viruses. They are expressing those receptors for their own purpose, for own physiological functions. But viruses can take advantages of those. So like influenza, they can react with or interact with sialic acid. This is a glycoprotein. Measles virus can take advantages of binding with CD46, and herpes simplex virus also can have advantages of heparan sulfate sugar. And rhinoviruses can use VLDL receptor, ICM receptor. Dengue virus can use these types of receptor. There are several types of receptor here, you can see. And so what are the evidences that the cell surface molecules can work as a virus receptor? There should be some receptor, there should be some evidences. So what are the evidences? There are four evidences here. One is, first, production of a panel of monoclonal antibody against cell surface protein. You know what is the monoclonal antibody?
Yes, sir.
Okay, so it has been generated from one immune cell against one type of epitope of the antigen. Okay, so production of a panel of monoclonal antibodies against cell surface protein. So if one antibody blocks the virus binding and infectivity, then the corresponding antigen is the receptor. So what is what is the meaning of this? Understand what they wanted to say here, or do I need to explain again?
No, sir.
Okay, that's great. So secondly, first we have generated monoclonal antibodies and incubate the host cell with those monoclonal antibodies, and we find those monoclonal antibodies binded with cell surface receptor and block the cell surface receptor. So when virus added with the cell, those cell will not interact with those virus, because the binding site of the cell that can interact with the virus has been blocked by monoclonal antibodies. This is the message from this. And secondly, soluble derivatives of the host cell surface molecule block virus binding infectivity. This is the blocking of virus receptor. So what we did, we blocked the viral receptor by what? By cell surface molecule. We generated different derivatives of host cell surface molecules and incubated with the virus, and those molecules recognized by the virus receptor and binded with those, and those viral receptor has been blocked. And when you add the cell with those virus will unable to bind with those cells because their receptor has been already blocked. And we can also do the same thing, the normal ligand for the particular host cell surface molecule that can block the virus binding, that is blocking the host cell surface receptor. একটা আমরা অ্যান্টিবডি দিয়ে আর একটা ভাইরাস রিসেপ্টরকে ব্লক করলাম, আর একটা হচ্ছে ভাইরাসের লাইগ্যান্ড দিয়ে হোস্ট সেল সারফেসটাকে আমরা ব্লক করলাম, বা যে কোনো লাইগ্যান্ড দিয়ে হোস্ট সেল সারফেসকে রিসেপ্টরকে আমরা ব্লক করলাম। এটা একটা। তারপর the number 4, introduction of the gene coding the susceptible host cell surface molecules into virus resistant cell, and the expression of that gene makes those cells susceptible to infections. Understand what they try to say here?
Yes, sir. Said they used a gene that encodes for a surface receptor that is like specific for the virus.
Yes, yes. So they have generated virus resistant cell. Those cell does not express any types of cell surface molecule. Okay, so those cell will not bind it with the virus. This is the first experiment. And secondly, they try to express the gene that encodes the cell surface receptor and incubate with the virus, and then the virus binded with that. But previously the virus cannot recognize the cell because of the lack of the host cell surface molecules. Okay, any question from others?
Sir, what is the difference between the first one and second one? It's more or less the same thing.
No,
right?
not something. The first one they use viral. Let's say the first.
I mean number 1 and 2. 1 and 2.
These two.
One is monoclonal antibody, okay, and then they again said soluble derivative.
No, those are two different. This monoclonal antibodies has been used to block the cell surface receptor, and those soluble
Yes.
derivative has been used to block the virus receptor that express on the viral cell surface.
Oh, okay.
Okay. Any question from others? Okay. So the forces that bind the virus attachment site to a receptor. The forces that bind a virus attachment site to a receptor might be hydrogen bonds or ionic attractions or van der Waals forces. Sugar moieties on the receptor or on the viral attachment site are commonly involved in those bond formation. Sugar moieties, that means the glycoproteins involved here, and glycoproteins have the glycans molecules. They interact here. No covalent bonds are formed between virions and receptor. So that means no covalent, no electron sharing, just hydrogen bond and ionic bonds and van der Waals forces. Initially, a virion is weakly bounded to a cell at only one or few receptor, and the attachment is reversible, and the virion may detach. But if this binding remains, that means if it remains attached, there are opportunities for more viruses attachment site to bind to more receptor on the host. So if sufficient binding, then the attachment to the cell becomes irreversible. Initially, a few number of viruses attach with the host cell surface and it is reversible. But when it's remain attached, then it will facilitate the binding of more virus with the host cell surface, and that become irreversible. And endocytosis, a cellular processes in which substances are surrounded by an area of the cell membrane, which then bud up inside to the cell to form a vesicle containing the ingested substance. This is called endocytosis. So endocytosis means the engulfing something by the cell. There are three different types. One is phagocytosis, and you can see, you know what is phagocytosis. Okay, the cell can eat anything. And pinocytosis, the cell can drink. That means extracellular fluid can be inserted into the cell as a vesicle. This is called pinocytosis. And another is receptor mediated endocytosis. So there should be something that will attach with the receptor expressed on the host cell and will internalize. So there are three different endocytosis process through which cell can internalize different outside molecules, and it is a form of active transport. That means energy consuming process. Okay. So endocytosis pathway can be subdivided into four categories. One is receptor mediated endocytosis, pinocytosis, phagocytosis, and there is another is caveal or clathrin mediated endocytosis, in which it is used by the cell for a variety of functions, including nutrient uptake and defense against the pathogens. So here we can study the receptor or clathrin mediated endocytosis. So cytosolic protein clathrin force the membrane. This is a receptor. When ligand bind to the membrane receptor, the cytosolic protein will force the membrane. These are cytosolic protein clathrin. Those are available in the cytosol, but when the receptor bind with the ligand, those protein will migrate from cytosol to nearby plasma membrane and force the membrane to bend around the virion, forming a pit, which is pins off to form a clathrin coated vesicle. Coated pits can concentrate large extracellular molecules that have different receptors responsible for receptor mediated endocytosis or ligands. That means low density lipoprotein, transferrin, growth factors, antibodies, and many others. And when vesicle is formed, vesicle loses the clathrin coated and receptor and ligand separated, and clathrin return back again to the cytoplasm. And here the fifth step, the receptors and ligands are separated and receptors are recycled to the cell membrane again to trap another viral particles, and the rest of the part will call late endosome. This late endosome will fuse with the lysosome or Golgi complex for degradation. This is the steps of receptor or clathrin mediated endocytosis. So if you are asked to draw the clathrin mediated endocytosis, you have to draw the figure with steps indicated here. Clear?
Yes, sir.
And now is looking for receptor mediated another endocytosis. This is called caveole mediated. So why it's called caveole? Because it is making a cave like structure. Okay, so caveoles are basically plasma protein plasma membrane bud which exist on the surface of many but not all the cell. They consist of cholesterol binding protein caveolin. The yellow color is cholesterol and this blue color is caveolin. So this caveole is made of cholesterol binding protein caveolin with a bilayer enriched in cholesterol and glycoprotein. The caveole are flux-shaped pit in the membrane like a cave. It is abundant in smooth muscle, fibroblasts, adipocytes, and endothelial cell. Uptake of extracellular molecule is then specifically mediated by receptor in caveole. এই যে receptor bind করলো, pit form করলো. Then phagocytosis is the type of receptor mediated endocytosis in which small molecules are transported across the membrane by caveole or deposited directly into the cytosol. So firstly, receptor will bind with virus, make a caveole like pit, make a cave, and then forming this, and then it will fuse the whole product with lysosome for degradation. This is the caveole mediated endocytosis. So where the GTPase dynamine plays a very important role to make a cave. So here, this is the dynamine, and you can see here, this is the part. So this is the dynamine that involved in dynamic closing and opening of the caveolar mouth for vesicle transport process. And the acting binding protein filamin A, this plays a very crucial role for trafficking the caveole. Okay, Achha. This is the acting binding protein filamin, and this is the caveole. It will help them to travel. So ডাইনামিনের কাজ হচ্ছে জাস্ট ক্যাভিওলার মুখটা খোলা বা বন্ধ করা, আর অ্যাক্টিনের কাজ হচ্ছে এটাকে ট্রান্সফার করা কোথা থেকে, সেল সার্ফেস থেকে সাইটোপ্লাজম তোমার আর্লি এন্ডোসোম ও লেট এন্ডোসোমে। আমি যেটা মোনোরেল সিস্টেম তোমাদেরকে পড়িয়েছিলাম সেরকম। And finally, the pinocytosis. Pinocytosis refers to the uptake of extracellular fluid and dissolved solids such as fat droplets, vitamins, and antigens. So usually it occurs from highly ruffled region of the plasma membrane, and it is a invagination of the cell membrane to form a pocket. So this is the pinocytosis. So here, this is the part. So this is the plasma membrane, and it is invagination of the cell membrane to form a pocket. This is the invagination of the cell membrane and finally form a pocket, which then pins off into a cell to form a vesicle filled with large volume of extracellular fluid and molecules. This is the way how cell can drink. The filling of the pocket occurs in a non-specific manner, and the vesicle then travel to the cytosol and fused with other vesicles such as endosome or lysosomes. Then phagocytosis. This is the process, and it is a very specific and depends on the cell being able to bind to the item it want to engulf by the way of cell surface receptor. So that means they use cell surface receptor, and it also depend on the material to be engulfed. It internalizes particulate greater than 0.75 micrometer in diameter into membrane-bound vesicle called phagosome, targeted to lysosome for enzymatic degradation. Those processes involve larger membrane areas than clathrin-mediated endocytosis or caveol-mediated endocytosis pathway. Phagocytosis is greatly enhanced by the attachment of a specific immunoglobulin G or complement fragment C3 to the target. তাহলে when a phagocytosis occurs through IgG and C3, that will be much more faster, and complement receptor like gamma Fc receptor of Ig or primary receptor is the phagocytic cells. Okay. So that is the important part, how the cells are engulfing something from the cell surface. Okay.